Hello everyone. I am Dibyadeep Chatterjee, PhD scholar at Wildlife Institute of India, and today I'm going to talk about long-term monitoring of two major cervids in a human-dominated semi-arid landscape of Western India. The study was undertaken under the supervision of Professor Kamar Qureshi and Dr. K. Shankar. So let's start by giving a brief account of the two cervid of a study. First, the sambar, the largest Southeast Asian deer, found widely in the entire Southeast Asia, and cheetah, the third largest deer, found widely in Indian subcontinent. Both these species are found in a wide range of habitats, and both of them are generalist feeders. They graze and browse according to forage availability. The conservation status of sambar and cheetal respectively are vulnerable and least concern according to IUCN, and according to the Wildlife Protection Act of India, that they are grouped under Schedule 3. Major threats to this species are poaching, habitat fragmentation, and encroachment. Moving on to our study area, the Sariska Tiger Reserve, located at the foothills of Aravallis in the Indian state of Rajasthan, encompasses an area of about 1,200 square kilometers, of which 400 square kilometers is a notified national park. Our intensive study area was 274 square kilometers of this notified national park. The forest type of our study area corresponds to northern tropical dry deciduous forest and northern tropical thorn forest. Sariska houses 31 villages, which has a human population of about 8,500 and a livestock population of over 30,000 that includes buffalo, cow, goat, and sheep. The people here are traditionally pastoralists. The rationale of our study is based on the fact that ungulates are primary consumers of forest ecosystem and act as a bridge between the primary producers and secondary consumers. Therefore, a long-term evaluation of their population becomes really crucial for the management and conservation of the ecosystem as a whole. With this rationale, we monitored the population density along with the grouping pattern and sex ratio of two major cervid in the semi-arid landscape of Sariska from 2011 to 2015. Moving on to the methods, we use line transit sampling to estimate the density, grouping pattern, and sex ratio of these two cervids. And the year-wise total effort from 2011 to 15 varied from 181 to 357 kilometers. And the data was analyzed using distance 6.0. Moving on to our results, the population density of sambar and cheetal respectively was found to be 14 individual per square kilometer and 26 individuals per square kilometer. And here it represents the population density of this tooth cervid during the entire duration of our study from 2011 to 2015. And as you can see, there hasn't been much change. There has been significant change in the population of both those species. Now moving on to the sex ratio, the male to adult female ratio of sambar and teethal respectively was found to be 30 males per 100 females and 38 males per 100 females. And the young to adult female ratio was found to be 18 per 100 females and 12 per 100 females for sambar and teethal respectively. Moving on to the grouping pattern, we observed that 71% of the encounters of sambar were found in a solitary or small family unit, whereas 70% of the teethal encounters were found in small to medium groups. Moving on to our inferences, the population density estimated in our study pertaining to both the species were comparable with the findings of other studies in similar landscape. The adult male to adult female ratio was found to be biased towards female, and the young to adult female ratio showed a growing trend in both the species. The grouping pattern was as expected to occur in the wild population of the cervid and is corroborated with the findings of other studies in similar landscapes. The tiger was locally extinct from Sariska and they were recently introduced. Therefore, long-term monitoring of the ungulate population becomes really crucial and key for the management of this endangered species. Studies based on scat analysis and kill records in our study area have showed that sambar and cheetal are important prey base of tiger, and with sambar being the principal prey species. Here it shows the results of tiger prey selection based on scat analysis and kill records. Camel trap study have also showed a high spatial overlap of tiger and sambar, and a high temporal overlap between both sambar and cheetal, therefore further proving my point. With this, I would like to conclude by saying the long-term monitoring of this of this cervid species are really important and crucial, not only for the long-term management, sustenance, and conservation of the endangered species, but also conservation of the habitat as a whole. With this, I would like to acknowledge the following people and ATBC for providing me this platform to present my study. Thank you very much.